Hi, I'm Rebecca Abraham. I was in the Yander lab this summer looking at aphids and Arabidopsis. Specifically, I was looking at Mysis persicae, which is the green peach aphid, and an auxin synthesizing enzyme that they secrete into plants while feeding. I specifically looked at this because the Yander lab saw this auxin synthesizing enzyme in aphid-fed artificial diet. Um, and they saw this through LCMS. And so other studies also showed that 48 hours after aphids feed on plants, that there is this increased expression of DAO1, which is the gene that encodes for the enzyme deoxygenase for auxin oxidation 1, and 48 hours after aphid feeding in Arabidopsis. And so the hypothesis is that while aphids are secreting their auxin synthesizing enzyme, that plants respond by increasing the expression of this gene and then oxidizing auxin. And this is because that auxin is a plant hormone and it's vital for plant growth and development. And so I was interested in seeing, well, how does this work in plants that don't have DAO1, so DAO1 mutant plants? And I would expect to see this increase in auxin in plants that can't oxidize auxin after aphid feeding. And so this is what I did. I did gust staining. And surprisingly, I did not see this increase in auxin, which would be relayed by a blue color, like over here in wild type plants. And that was really surprising, and that would need further investigation to kind of see what's going on there. And then I also did aphid choice assays. So I wanted to see if aphids would prefer feeding on plants that can't oxidize auxin or just regular wild type. And so I gave them 24 hours to choose between DAO1 leaves and wild type leaves and to see if there's a preference there. And surprisingly, there was actually no preference there as well. We kind of thought because there's this increased reproductive rate of aphids on DAO1 that they would prefer DAO1 plants, but there wasn't a significant difference between the two. And so moving forward in the future, I would definitely like to repeat these just to confirm my data and results, especially the gust staining, because again, this was pretty surprising not to see this increase of gust activity in these DAO1 plants. And also the aphid choice assays, I would kind of play around with the experimental parameters, probably increase the amount of hours to 48 hours, just because the induction of DAO1 really does happen after 48 hours. And yeah, that's what I did this summer.
Do you have a hypothesis as to why in your mutants the GUS did not accumulate?
What I'm thinking is there are other ways to catabolize auxin. And so I'm thinking that Arabidopsis probably increases the expression of those genes that controls the catabolism that way. So maybe like amino acid conjugates or glucose conjugates, and that way, yeah.
Great, thank you so much.